My presentation is about a cell-free bioassay for the non-targeted detection of designer androgens. So in the labs now, if you want to detect an androgen from an athlete's sample, you need to know its structure because mass spectrometry is used based on the fact that androgens have different structures, and on mass spectrometry, they give a different spectra that allows for their specific identification. Now, the problem arises for designer steroids for which there is no known structure, and therefore the identification may not be so easy to obtain. And so at present, if designer steroids are thought to be in a sample, actually a complex and expensive suite of analytical testing is performed. Such testing doesn't lend itself to high throughput analysis for biological passport samples or even race day samples. And so what is needed is a high throughput, non-targeted test for the detection of designer androgens. And in my lab, we have developed such a test. So we don't base our test on androgen structure, but on androgen biology. And so when androgens act in your cell, they'll diffuse into the cell and bind to the androgen receptor. The androgen receptor will then dimerize and enter the nucleus, where it binds to a specific DNA element called the androgen response element. And the androgen response element will lead to the activation of gene transcription, and this then leads to the androgen cellular response. And so all androgens, whether they're your endogenous one, testosterone or dihydrotestosterone, or anabolic steroids or selective androgen receptor modulators, SARMs, doesn't matter what their structure is, and this represents a vast variety of different structures. Doesn't matter about the different structures, they all do the same beautiful thing of activating the same biological pathway, which we are able to exploit. And so in the lab, we've mimicked this biological pathway and developed an in vitro androgen bioassay. But the thing that we've done is we've taken that biology that's inside the cell and we've taken it out of the cell, so no cell biology is needed. And instead, this whole assay can be done in a 96-well plate. And so here, androgens will again activate the androgen receptor. This will then bind to an androgen response element on a DNA transcript that will lead to the synthesis of a fluorogenic molecule that we can easily measure. So the whole process takes place in 1.5 hours, and because of that 96-well plate format, we're able to measure a number of different samples, many different samples simultaneously. So we're developing what we can see as a high throughput screening approach. So to show that it works and it's sensitive enough to detect biological activation of androgens, here we show a testosterone dose response curve. So this is a testosterone dose response. Higher testosterone concentration leads to higher levels of fluorescence. And we see in EC50 around 0.4 nanograms per mil. But our assay just doesn't detect testosterone. We've shown it to detect all of these different structures. So it can detect a number of different SARMs, it can detect a number of different anabolic steroids, including some of your original designer steroids, and it can also detect testosterone in its different formulations. And so this is great in showing it's a non-targeted androgen test. But all of this data is with androgens that are in their pure form dissolved in ethanol. So we also wanted to make sure that we could detect these anabolic steroids when they're administered to a horse and we're provided with a biological sample. And so here we were given the plasma from an administered horse that was given oxyguno, or a different horse that was given 6-oxo. And at these earlier time points, you can see an enhanced androgen bioactivity, which was told us that this horse was had higher androgen bioactivity, indicative of the fact he was given these anabolic steroids. So we can also detect these in the in vivo situation. So we've also used this assay just to screen the out of competition samples from 80 different resources. And normally the information that the analytical lab would get would be the testosterone level. We see this rather one-dimensional set of data, low levels of testosterone all the way up to high levels of testosterone, in keeping with colts, geldings, and mares. What we did was then add a second dimension to this same set of data. So we still have our testosterone going from low to high, but now what we've added in is androgen bioactivity from low to high. And what we see is there's a good correlation, low testosterone activity, low androgen bioactivity, up to high testosterone levels, high androgen bioactivity, with a correlation of around 0.7. Now, in these 80 samples, we found one anomaly where it had low testosterone levels but high androgen bioactivity. So in this way, you can see that we show that our non-targeted androgen bioassay can pick up or red flag a particular sample. So in this way, the test would complement the testing that's currently being done in the laboratories for targeted androgens, and therefore we will actually add to the arsenal of the anti-doping screening. Thank you.